Hey, science students. Welcome back. Hey, welcome back. Hey, thank you for opening this up. Hey, thank you for being a part of this connecting in this way, getting on your educational journey. Thank you so much. Appreciate you doing this. The, the first thing you might be saying, whoa, you've already got all the lines and the boxes and the arrows and everything. Usually you draw those as we're going, and that's true, and there's a good reason for that. This is take number 2 on making this video. Yeah, we made it, and we finished recording, and when you click on this little red button over here to say stop, and you can see there, what happens is in the Loom server pops up and it processes it. In other words, it cleans it up and gets it all ready for viewing, and that takes about 10-15 minutes to do. And in that process, and while I was recording the other one, we had a big storm come through and lots of loud thunder and lightning, and in that 15-minute window when it's processing it, the electricity flipped and shut down. Of course, that shut down the computer, and you know what happens to information on websites that is processing, boom, gone. So, grumble, 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 complain, complain, complain. And so here I am, round 2. So I didn't go back through and erase everything. So this will probably be a little quicker, which you're going, yay, for that. A little quicker because I don't have to do all that stuff. And but I do want to share the information with you in a good, professional, high quality manner. So with that, let's get going. Again, welcome, Nevada High School students, Crowder Bio students. Welcome, welcome, welcome. Oh, another good thunder roll. Oh, if that happens again, I don't know. Guess we just have to wait until the storm ends completely. But we're going to hope that while we're talking, it clears out and goes from there. So let's get going, and fingers crossed, fingers crossed, fingers crossed. Hey, so we're going to start with the nucleus. I mean, the main player. And as we go through here, one of the things that we want to do is metaphorically kind of give you a connection to what they have. But all this stuff you had as in your bio EOC class. And so if you're a recent high school graduate or you're in high school now, this stuff you would have gone over in that class, so it should be complete review. So we'll just roll through it in a fairly quick manner. Doesn't need to be taken too long on things that you have had. If you're a junior at Nevada High School, it was just last year that you had it. So if you are a senior 2 years ago, if you're a bio or if you're a Crowder student and graduated in 18, 19, 17, somewhere in there, fairly recent for you as well. So here we go. And my lights keep flickering just a little bit, so I'm pretty nervous about this, but here we go. Nucleus, okay, genetic materials in there, DNA, that's where the duplication happens, the replication happens. But one thing we want to think about is that it is a membrane-bound organelle, okay, and you can see here on my line what that simply means. That's the edge, that's the membrane. That plays a big part in what goes in and out of the cell, or the organelle, I should say, not cell, as with all membranes. And this is the image of it down here, the plasma membrane, that kind of membrane edge right there. Some other key components of the nucleus, the nucleolus, okay, that's this big darkened circle that I've got highlighted here, okay, and you can see here, that's where RNA takes place. RNA synthesis takes place. Super important, super-duper important, and the things that help make the RNA, that's where they get made. Everything's got to start somewhere, and that's where they start. Our next huge important organelle to talk about, the ribosomes. Okay, the ribosomes. First thing to recognize, 2 parts, You've got your large subunit, this orange part here. You've got the white, the small subunit. Those are two individual parts that come together to form the ribosome. And pretty cool process. The RNA strand moves through here, and this tRNA reads those codons and associates the correct amino acid, which forms a protein, and they're all connected by connected by a peptide bonds. And hopefully you can see why this is called a tRNA. It's got that. Oh, didn't mean to do that. I'll have my ink tray up. Oh, in case you didn't notice, I'm using blue today. Got that little T frame right there. So that's the ribosomes. It is a huge player in the protein production game. So that is the ribosome. So let me close my ink tray here. Actually, you know what? I probably won't need the ink tray at all because I've already done all that stuff. We're going to slide up here to the plasma membrane. Plasma membrane is super important. Phospholipid bilayer. You can see those right here. Here's one phospholipid. Here's another one. So there's two sets. That's why it's a bilayer. But I am going to open it up just in case. I do like to have it up. Bilayer, two layers, tails coming together, polar heads on the outside. We talked about phospholipids in the protein section. No, not the protein section. The lipid section of the macromolecules. Its function to control what comes in and out of the cell. Super important. Chapter 5 is all about the plasma membrane and everything that it's going to do for us or does for us. We will detail that in heavy detail fashion in Chapter 5, which is your next chapter. And as always, high school students, these boxes with the check mark, be sure that you've written those down. That's what we're going to be looking for when it comes to the notes. So be sure that you have taken care of those. So let me close this ink tray and move this up a little bit more. Okay, our next organelle to talk about is the ERs, the rough ERs. Rough endoplasmic reticulum, smooth endoplasmic reticulum, both important. Both have functions of their own that they do. And the rough ER is simply a reference to having the ribosomes attached to them. And you can see the nucleus here. This one they chose to make it yellow. Typically see it purple, dark color. Excuse me, but when they go yellow with this nucleolus here, to me it looks like an egg, and that's the yolk there. But this section here, the rough ER, smooth ER, the rough and the smooth are reference to ribosomes. Either it has ribosomes, doesn't have ribosomes, so it gives it that smooth or bumpy look, whichever way, and that's why the names are as they are. But the rough ER, RER, that's all about it has proteins or ribosomes, so it's making the proteins. The smooth ER does not, and that's where lipids are made. That's where lipids are made. So super important there. I mean, obviously we don't want a lot of fatty acids, but we do need to have a certain amount of them, just like with cholesterol in the plasma membrane. That helps the fatty acid tails kind of stay in line or stay the shape they need to. Those things are in large quantities bad for us, but in modified quantities, we need them. They're helpful. We got to have them. Okay, so that's the reticulums, the endoplasmic reticulums, rough and smooth. Our next player in the protein production is the Golgi apparatus, or it's also referred to as the Golgi bodies. I wish scientists would just pick one name for stuff and go with that, but that's not the case sometimes. But again, information here. High school students, be sure you collect that and have it ready for me. Now, the way the Golgi apparatus works is the proteins are made off the rough ER, and then they're put into little vesicles here, which is really just part of the membrane of the organelle. And when we get to chapter 5, towards the end, we will showcase exocytosis, which means it's the process of the material, whatever it is. In this case, it's a protein. As it exits that organelle, it goes through that membrane. That membrane kind of just folds around the material and kind of buds off. And then the membrane reconnects to cover up that empty space. And then just makes more phospholipids and gets more, and then rebuilds it and puts it back to its normal length. But the incoming vesicle just kind of enters the Golgi apparatus and just makes its way through these interconnected membranes or sacs. And from the top here, it looks like just a series of these rubber bandish looking loops. But what you can't see is that underneath, on the backside, it's all connected together. And they just make their way through. And when we think about the Golgi apparatus, we're thinking post office. We're talking FedEx. We're thinking UPS. Excuse me. We're thinking those things with the idea of preparation, distribution. If you think about, say you wanted to ship something somewhere, you could actually walk into a FedEx office, a UPS office, post office, and all you got is the thing that you want to ship. And you can get a box, you can get the materials inside, the packing peanuts, the bubble wrap, whatever. They got tape, they got stickers, everything you need to get the package ready to go and ship it out. Golgi apparatus. That's what its process and all this. And again, when we get to chapter 5, we'll talk about exocytosis, which is what's going on here, and then we'll refer back to this process to remind you that's what's going on here. All right. This next section is a review of the last 3 organelles, but they're bringing all that protein stuff into one section here. So again, high school students, make sure you collect this. But this 3-part process of making proteins, the first part is the ribosomes, building the proteins, the rough endoplasmic reticulum providing the vesicle, the transport vesicle, and this is like a little protein in here, and it buds off the membrane. Exocytosis, again, a full detailed explanation of exocytosis is coming in chapter 5. So, but again, we will refer back to this as a reference point. And then it enters the Golgi apparatus, as we just talked about, making its way through. And then when it gets to the end, the transport vesicle. And in this case, it's actually leaving this area and headed out to the rest of the cell. So that is the 3-part, the ribosomes, rough endoplasmic reticulum, Golgi apparatus, the 3-part protein making process. Our next organelle to talk about, the lysosome. Super important. Think trash people. I shouldn't say trash people. Think trash men, trash women, and not in a negative connotation, in a positive connotation. Their job, their service they provide is super important. We would be in a mess if we didn't have them come along each week or whenever and take away the trash that is collected in our daily lives. In our daily lives, super important. Just like the lysosome. The lysosome, you can see, it's got these little pseudopods, and it just kind of motors around the cell. And when it finds some broken down stuff, some old stuff, some waste. Even if it's a um, foreign matter like a bacteria or a virus or something, it gobbles them up. You can see here it actually takes it in and breaks it down with digestive enzymes. Think stomach acid, breaking down food. And when that happens, it dissolves it, breaks it apart, and then ejects it out, and as it prepares for it to leave the cell. Okay, leaving the cell. So that's the lysosome. Getting rid of all the refuge that we have around. Okay, our next organelle is the vacuole. Okay, vacuole is super important. You're thinking storage. Okay, you're thinking the city water tank. Okay, you think about those water tanks that cities have. If it's a smaller town, maybe you just have one. If it's a little bit larger town, maybe there's one or two or three. Big cities would have a whole bunch of them sprinkled around. But you're thinking those water tanks. Maybe it's got the school mascot on it. Maybe it's got a fancy slogan. Maybe the city has a symbol or a map. It's got that on there, something recognizing that belongs to that city or that's part of that city. All right, but they come in three styles, three types. We're going to talk about the central vacuole. That's what's here in front of us. This is the plant cell. Got your chloroplast, got your cell wall. It'd been nicer if it was a little bit more rectangle, but it is what it is. But anytime you see the chloroplast, anytime you see a cell wall reference, that's your chloroplast. And in the plant cell, it's got this big vacuole. And when that thing's full of water, and I drew these little arrows here, when that thing's full of water, it presses against the sides of the cell wall. And when it does that, it creates turgor pressure. That's what this word is over here. And again, we'll talk about that more in, I believe it is chapter 5 that comes up again. But maybe it's, probably chapter 5. Turgor pressure. It's that pressure that's created when the vacuole is full and it's pushing up against the cell wall, and it kind of acts like a vertebrae. When you stack those cells on top of each other in a plant, just like our vertebrae formed our spine, it gives the plant the ability to stand upright. Okay, makes that possible. And so that's why when you neglect plants for a little bit, or they're outside and it doesn't rain in a while, they start to lose that water, and then they wilt over or lean over because the vacuole is getting smaller because there's not as much water in it. But as soon as you water the plant or it rains outside, then that vacuole fills back up and the plant stands back up. So that's the central vacuole in a plant cell. Storage vacuole, that's us. Okay, animal cells have storage vacuoles. So instead of having one big one, we'll have lots of little ones, okay, little circles, and we're just putting some water in there. I was kind of storing it for when the cell needs to use it for all the processes, cellular respiration, cellular respiration, DNA duplication, whatever it may need oxygen or hydrogen or water for. So little storage vesicles, vacuoles all over the cell. The last type is probably maybe the coolest type because it's part of the locomotion of some fungi and protists. Protists, if you don't know, are these little microscopic organisms that live in all kinds of mediums, typically water. If you were to go get you a cup full of pond water and we took a drop of that and made a microscope slide, you could see the protist in there. There's all different kinds of types, but little microscopic organisms. And actually, where you do see protist-like organisms, if you think if you think in movies, whenever they show these little microscopic things kind of floating around, and one of the movies that did it a bunch, that was the Ant-Man. When they send them down into, lost my word for where they are sent to, like their wife is there, and Paul Rudd goes there a couple of times. But you see those little things floating around, and a couple of times one of them tries to attack Michael Douglas's little ship that he's in. Those are protist looking like organisms, kind of where they're getting the idea of that from. But protists, these microscopic organisms, and you can find them in pond water all the time. They're pretty cool looking to see. But that's a protist. They have contractual vacuoles. So for contractual vacuoles, think blowholes on marine animals like a dolphin or a whale or things like that, to where they take in all this water and trying to get some food. They filter out the water, they eat the food, and then the water is ejected through the blowhole. Okay, protists do this with their contractual vacuole, and when they do that, because they're in water and they blow that water out, it creates a Newton's third law situation to where there's an opposite reaction for every reaction. So when they throw the water out, it pushes them the opposite way and they move. It's one of the ways that they move. So pretty cool concept there. So that's the vacuole. Now let's move to our next two organelles that we're kind of going to do simultaneously, because they're doing their functions are the opposite ends of this great cycle, the energy cycle, where you've got the making of the sugars, the breaking down of the sugars in photosynthesis and cellular respiration. Excuse me. So we'll talk about them kind of simultaneously here. And again, I got this box here, check mark to remind high school students, be sure to have this information in your notes. So mitochondria is the orange one here, obviously. Chloroplast is the green one because of that photosynthetic materials, the chlorophyll reflecting that green, but some structure stuff. The mitochondria has got these 4 spaces, 4 sections, and when we talk about cellular respiration in Chapter 7, we will detail these because as cellular respiration is occurring, each stage we go a little bit deeper into the mitochondria. Start at the outer membrane, go inside, and then we go into the inner membrane, and then into the matrix, this salmon-colored material here. But each stage goes a little bit deeper. But again, Chapter 7, we will talk about that a bunch. Chloroplast. Okay, some basic structure here. You've got your stroma, which is kind of this cytoplasm. This is light-colored section here, all through here. That's your almost similar to the cytoplasm of a cell. It's just kind of a gel-like substance. It kind of holds everything in place, keeps it nice and safe. You've got your thylakoids. That's each individual coins or token-looking things. That's a thylakoid. If there's a stack of them, okay, that's a granum. Okay, so this would be a granum here. But if I talk about all these stacks, then that's a grana, collection of them. But the idea here is sugars are made, photosynthesis, which are so important. I mean, because without it, okay, we couldn't survive. Okay, we've got to have those sugars. That's what our food is. So super important. Our next topic, and I think it's our next to last topic. Yeah, we got the microfilaments, and then we got communication, and then we're done. We're wrapping it up. Okay, as always, be sure you've got this information in the box here. So let's talk about these filaments here. Now, hopefully these that I have this box around, when you see the microtubules, I hope an image jumps in your mind pertaining to mitosis. When we get to metaphase and the chromosomes are moved into the middle of the cell and lined up, almost like a square dance partner connection there. And I say square dancing, that may not even be a relative term. You may be sitting there thinking, square dancing, what are you talking about, man? Look it up, you'll see. Google it. The microtubules are the ones that move them to the middle, and then when it's time, the old anaphase, snapping apart, separating them to the opposite poles, those are the ones that do that. Okay, that's the microtubules. We've also got the microfilaments around the edge here. So certainly support support is a main part of that process. And then what may be, I should say most important, but they're all important. But the intermediate filaments, you can see here we go along the edge. Okay, so again, we're thinking support. And then these ones that are kind of waving all the way through the cell, what would be the cell, those have a couple of purposes. One, they kind of anchor everything in place. So if you're an organelle and you're kind of in this area, like if I get my pen up here, if you're an organelle and you're kind of in this area, then you're going to kind of be connected to that intermediate filament, and it's going to kind of hold you safely in place. Another purpose or function is transportation. Things can move along these threads as needed. They can move along there and be connected in that way. Took a little drink there of water. All right, let me close this, and then we'll talk about our last topic and the second part of chapter 4, and that's communication. That's what this COMM over here is for, communication. A couple things to talk about off the top. Glycolipids are part of the process that cells recognize the material. Is it something that we need? What is it? Is it us? Is it safe? The glycolipids are part of that process, and when we get to chapter 5, we will talk about that to death, a lot, about how the cell, the plasma membrane, the checks and balances that it has to make sure something that needs to come inside. Again, some other proteins that are within the membrane, the MHC proteins, they're responsible for recognizing, hey, is that something good? Is that us? Or is that something foreign? And if it's foreign, it don't get to come in. And that also comes into play when it comes to organ transplant. The MHC proteins, they're designed to protect us, but in the case of organ transplants, what may happen is your immune system is supposed to recognize, or its purpose is to recognize when something is not us and attack it. So if a person has gotten a lung transplant, a heart transplant, a kidney transplant, on and so on and so on, then the immune system is designed to see that and destroy that is its purpose. But you can't have your new organ destroyed. That's bad. So you've got to take medicine that represses your immune system, which makes it vulnerable to other things, and especially in the condition we're in right now with the COVID stuff. That's dangerous stuff. If your immune system is repressed, doesn't respond like it needs to, and yet we've got this deadly, dangerous virus out there. But this section is about communication, so let's talk about the 3 ways that happens. The first 2 are connection points, anchoring points. We've got these tight junctions. You can see them, they almost look like brads, like in upholstery or on jeans, decorative stuff, but also designed to hold things together. One thing I want you to recognize is that these 2 layers are the edges of 2 different cells, just like they are here, just like they are here, and that's identified right here, and then cell 1, cell 2. So these are these are 2 cells that are being connected, anchored together. And it can be tight junctions, as you can see here, or welds, or brads on jeans, or upholstery, you can think of that. The other way is anchoring junctions, and that's these little things in here. And of course this is intermediate microfilaments out here, but they can connect the 2 cells. Now once they get connected, then we go to this 3rd layer, and that's these connexons, Think of these connexons. If I got two houses that are side by side, and maybe there's two windows that are facing each other, if we open those up, you and I can talk to each other fairly easily. We're probably just a foot or two apart. And if you need something, I can hand it to you. If I need something, you can hand it to me. But we can communicate. We can share things as much as we want. They can be open or closed. You can see here, if they're closed, nothing's happening. But if they're opened, because it goes all the way through. Okay, if one's open, they're both open. It's one thing. And then we can exchange or communicate or whatever needs to happen through there. But tight junctions, anchoring junctions, communicating junctions, that's our way of communicating with the cells. One example of how this happens is when a cell gets infected by a bacteria, it sends out a message, hey, I'm in bad shape here. And then the other cells shut down, close the walls, close the gates, pull up the drawbridge, all that stuff, and until the immune system, the T cells come in, the macrophages come in, and clean it all up, and then they open back up for business. So, just to show you here, that's the last of the material, and that's probably a little quick because they didn't have to draw, or I should say, not quick, but less time spent because they didn't have to do all the drawings and all that stuff and emphasize that in that manner. But again, thank you. Love you guys. Appreciate you guys. High school students, make sure you collect everything, all that information. That's all I'll be looking for when I check notes when it's time. College students, that's completely your call. The more you write, the more you'll learn. The more you'll know, the more you'll understand. So it's Saturday, you can see there, August 29th. So for high school, it was the end of our first week. For college, university, or Crowder, into that second week. But when you watch this, for Crowder, it'd be that fifth week. We had 1 week for a test, and so fifth week. So we're deep into possibly even getting ready to have October. So a little bit cooler time. But for high school, this is chapter 4, and we're doing 9 units. So we're close to halfway. So we're probably post Thanksgiving, staring that Christmas break in the face. That's going to be awesome. So thank you for watching this, and I hope you're safe and sound still, and everything's going all right. And with that, we will say goodbye until next time.